David Scott, and talking about heterogeneity and ion binding for bacterial antimicrobial resistance and some good ideas.
Right. Thank you very much. And thank you for the invite, and apologies, I have to do this from a seated position at the moment. So, what's the problem? I just wait for everyone to pause a second. Yeah, don't worry. Just rang the bell outside. Right, okay. So I'm kicking off the soft matter and biology session. I'm a biophysicist by background, which means I've worked between both the biology and the physics field for getting on for 30 years now. So it's really nice to be at this conference and have a bit more soft matter and physics in because I normally exist in a microbiology department where they do maths, but usually it's in orders of 10. Right. So I'm going to talk to you today about a project area which there's a bunch of us trying to draw this together, where we're trying to look at sort of this bacterial system over sort of various levels of complexity. And I think a lot of the things and the techniques that have been talked about this conference actually would be quite good. So this is trying to bring together some rather disparate bits of projects area. It's been going on for about 20 years into one cohesive whole. And the problem we're looking at is ion binding, which seems a bit esoteric, but as I'll go towards the end, you'll find out actually that has a real, very, very practical application, clinical application. And the idea behind it is basically that bacteria take in ions to do various jobs and also encounter environments where ions are quite mixed and some are toxic. And how do they cope with that? Or how do they try and predict the future, essentially? So, this work wasn't done alone. A lot of this work was the work of Karishma and Ryan, who were two PhD students in the lab, who did a sterling amount of structural and functional work on the proteins involved here. My main collaborators are John Hobman, who has been working on, he's a microbiologist working on ion binding and metals in biology and bacteria for about 30 years now. Another colleague of mine, Dov Steckel, who is previously of, I believe, Trinity here in maths, who's a mathematician by background, who models a lot of the flows in this as well. And Steve Carr, who's a structural biologist based at Diamond. And a lot of this work was done at the research complex at Harwell, which is next to the Diamond synchrotron, where we get to play with lots and lots of very big, nice toys. So, bacteria, very, very quick overview. If you take a mixture of bacteria and you put in something called a Gram stain, you'll see it tends to distribute into two colors, a dark one, which stains, and a light one, which doesn't. And we define these as gram positive and gram negative, and this is to do with the structure of the cell wall. Those that are gram negative have two membranes, and that's what we work on here, our model E. coli. And if you look at an EM, you can just about see, even at this resolution, that there are two membranes surrounding the bacteria. So we have an inside, we have an outside, and we have a periplasm. And at some point, things have got to get from the outside to the inside and from the inside to the outside. And there are many metal response systems within bacteria, and they seem to be, by necessity, quite promiscuous in what they do. So you'll have a system, for instance, with nickel, which also acts with palladium, copper and silver, ones I'm going to talk quite extensively about as well, but also zinc and cadmium as well. Both seem to react to this system, but the way that sort of biologists defined this in the past is basically to say, challenge a bacteria copper, it's resistant, it therefore has copper resistance, challenge it with silver, It therefore has silver, and therefore it's silver resistance. And what we're discovering is actually, when you start to look at the details of this and really drill down, is that these are actually, broadly speaking, the same thing. So the question becomes, how good are these systems at discriminating? Can they discriminate maybe across groups as well and react? And can we actually sort of define a sort of what sort of level that description of the bacteria, that discrimination is coming at? So why? Well, bit of a wordy slide, but that's because bacteria seems to operate, we look at these things at many levels. Molecular, so I can get the proteins, I can do structural analysis, I can do functional analysis, I can see how things bind, KD, rate constants, etc. I can look in vitro and then try and sort of relate it to the in vivo as well. Cellular, where you look at basically the arrangements of the components, and always popular these days with super resolution microscopy, where you can look at how things move, come together, and try and relate that maybe back to the structural work. More sort of above that is the population, where we're looking at the individual cells and how they act in a population, how they express different components, the heterogeneity that goes on there as well, which obviously is related to the cellular organization, which obviously is related in some way to the molecular. And finally, spatial, that these things don't just exist in a nice warm flask in your lab, out in the environment. They're going to be in biofilms. They're going to be on surfaces. How does the 3D arrangement of that affect things, and how heterogeneous is the responses to ions across all of these levels? And the sort of things that you start to talk about in active matter, for instance, are sort of very much related to this top level here, but the origins of them are very much related to the molecular level. And one of the problems, of course, is that, if you're a biophysicist or structural biologist, you've been messing around at this level. If you're a microbiologist, you've been doing at this level. And if you're a cellular biologist, you've been working about here. So you have a problem of basically transdisciplinarity, that not everybody's talking the same language or realizing actually the significance of the results in one area is quite significant in another area. So at the molecular level, it's probably quite easy to understand this heterogeneity. And I'm going to talk more or less exclusive now about the copper-silver system, because that's the one we've got a lot of results on. If you look at sort of the molecular levels of the ions, for the most common ions, you can see that silver and copper both have basically filled up d levels, d orbitals. And you can see how, if something was recognizing it, well, it's just looking for something that's about the same sort of shape. And indeed, these ions form the same sort of thing. Copper 2, for instance, does not form tetrahedral compounds. So you can start to see, there may be some sort of form of discrimination is going on there. So you've got something which you basically say this could be happening. And of course, at some level, you get sort of quite a level of complexity about how these things cope with these systems. And typically, we have copper systems which can operate either under low or high concentrations. But predominantly speaking, this is the inner and outer membrane. You have systems here which basically efflux things out, systems here which sort of chaperone things onto these systems as well. And these can be carried either on the bacterial chromosome or on mobile genetic elements, plasmids as well, so they can be passed around quite quickly. And the silver system and the copper systems, when you actually line up the sequences and you look at the structures, look remarkably similar. And one of the things that we've really drilled down on is these proteins CusF and CILF, particularly CILF, which are the chaperones which bind these ions and are supposed to convey these things to these large efflux pumps. And there's a few more results coming in as well, make us sort of start to reevaluate that and what's going on. Oops. And you obviously have other zinc systems as well, for effluxes. Now, one of the interesting things is when you actually look at these under the EM, and these are silver resistant E. coli, you see these little black spots here. And these are nanocrystals of silver, which are formed by the system. And you'll notice that the silver is not on the outside, it's on the inside. So when you have an efflux system, which is supposed to be fluxing things out, and it's energy dependent, how does this stuff end up on the inside? And again, that's a bit perturbing. And these are, you can separate these out, and they're silver metal. And not the iron. And we know that it's the iron that is actually, it has the bactericidal effect for silver. And we're interested in silver in itself from a microbiology point of view because it's a very, very powerful antimicrobial. It's more deadly to bacteria than mercury, but far more benign. You can't really drink mercury. You can drink quite a bit of silver, ions and colloids. Not recommended. You will go blue, okay? Because the colloids get under your skin as a photographic reaction, right? And there's a condition called aguria. And I did have some photos of somebody who actually done this, and literally they look like a Smurf. Okay, right. So looking at these sort of things is quite important because obviously we want to try and kill bacteria under certain conditions and keep them away from things. So the full title of the division I work in is Microbiology, Brewing and Biotechnology, and the brewers are very interested in keeping bacteria out of their yeasty systems. And so we had a look at the SILF protein and we solved the structure of it with silver and copper. And it looks remarkably like the CusF protein and its alpha fold prediction, except for this helix at the back. Right, and it has sort of, you can see where all the important residues are for iron binding. Okay, that's fine. That's standard structural biology. But the question becomes, how does it actually work? Well, when you actually drill down to have a look at the structure, you can see the silver ion sits in its binding site quite tightly, but the copper 1 ion sits there and has an extra water there. Oh, that's interesting. And you can do the MD, and you can see that the water in the silver is more excluded, but in the copper it's there quite often. Well, that's quite fascinating. We've looked at systems before where we have involved water, and we have effects such as enthalpy entropy compensation. We could be seeing something here where the thermodynamics of the system dictate that this is binding quite tight, but able to use water to have the same affinity, but different enthalpies and entropies. So we can do a bit of calorimetry, do some isothermal titration calorimetry. For those that don't know, you basically put your protein in here and you titrate in your ligand, and you get heat changes. You measure the heat changes here, and basic thermodynamics is that, that if you sum up the heats here and you know how much your concentration is, heat per mole is your enthalpy. If you can fit that, you can get a KD, dissociation constant, which is related to free energy. Use that, you can also get the entropy. So in one experiment, you can determine all the parameters. That's nice, and you can do it for copper and for silver. Of course, with the copper, we have to do it in anaerobic conditions because copper 1 forms copper 2 very, very easily with oxygen. And the values we get out are remarkably similar in an affinity, but the enthalpies and the entropies are pretty much all the same. So it's a bit strange. Okay, not operating how we think it is, but not entirely unusual. Also, the stoichiometry, the affinities we get are a lot tighter than we're seen for the other CusF system as well. So we do a bit of MD on this, and so you can just put it in a water box and move the protein around, and we see that indeed there are some parts of the protein which do move quite a bit. and it's to do with this helix here. And and you can look at either the copper, the silver, or the apoform, and there are changes in the rest of the protein, which is helping this bind. Now, and again, so the origin of this enthalpy entropy compensation of this very similar affinities didn't entropy compensation, but appears to be from the actual movement of the protein. And I say that's slightly unusual because we've been working on lots of other systems. Dipeptide binding protein from E. coli, it's from E. coli, it binds dipeptides, all 400 of them, with very similar affinities, but very, very different enthalpies and entropies. And obviously water is involved. We've looked at lipocalins, which are involved in odor perception, and titrated in various compounds. And there you see definitely there's release of ordered water, and the whole affinities of these things are very, very similar. So looking at systems which have a dropped specificity, but similar affinity, usually you're looking at enthalpy entropy compensation, but here we're not. So this was a bit of an outlier for us, but we can sort of start to explain how that works. So that heterogeneity from ion binding may well come coming from the proteins themselves being, if you like, designed, using that word with caution, of course, to take in these ions with very, very similar thermodynamic properties. And we also tried to look at another protein, SIL-E, which is interesting because it's what's known as an intrinsically disordered protein. It doesn't fold to a unique structure until you put ions in, and the whole thing just folds up into alpha helices like a sponge. And we can get it to bind under certain circumstances, all up to 8 silver ions, but also different ions as well. So these proteins will be binding different ions as well in a system which allegedly is only tuned for silver. And here we get, I think, to a semantic problem again, that when you look through the database, yes, you have a cluster of silver ions and a cluster of, silver resistant proteins and bacteria and a cluster of silver resistant, copper resistant ones. In fact, they are the same thing. It's just really what they've been challenged with first. And when we've gone through and we actually have solved the structure of SIL-C here, which is this membrane protein, all the important residues that actually do with ion binding are in exactly the same position and exactly overlaid with the cusp-C, the coupled version. And all the residues that are different just lie on the outside and could just be sort of small mutational differences, benign mutational differences. So the actual systems themselves seem to be very, very, very similar. So that's one of the major things we've done. And we can starting to look at how we can look at the cellular heterogeneity. And one of the ways we've done that is to try and label one of the proteins. So we've labeled SIL-C here with GFP, and you can see your nice bacteria. And if you look up close, you can see individual spots here. So we're now in a position where we have this system where we can actually start to look at these components coming together. And the idea is to start to label many of the other components as well with different colors of fluorescence and start to put these things together. Because it is known in the CUS system, where they've done similar single particle tracking, that this CUS-C seems to move around the outer membrane quite happily. You add the metal challenge and it slows down a lot as if it's assembling the whole complex. And they think it's assembling with the rest of this, but it could, of course, be assembling with this or any other export system out there as well. And as I say, the function of SIL-F is also to transfer this across here. And they measure the affinities of copper in CUS-F and in this system here. And it seems that this is a lower affinity, this is a high affinity, and therefore this thing is being effluxed out. However, our results show that this is of a higher affinity to what this would be expected to be, which means the system, what we need now is measure the ion affinity on this. Otherwise, the system is running backwards. It's actually taking things out as well. And that would lead to perhaps the intriguing result we have here, of where these things, these little nanocrystals sitting in the periplasm, and it is known that the nitrate system can actually take the silver ions and turn them to metal. And remember, the metal is actually quite benign and forms these little nanocrystals. And that seems to be what's happening there. Because then that would make sense that this thing is loading up and taking things from the environment and putting it there. And when you do the calculations of what they call the minimum inhibitory concentration, i.e. the minimum amount of silver you need to kill a bacteria, it's about 600 micromolar, and what the size of a nanocrystal would be if you got all of the 600 micromolar across the cell and put it into a nanocrystal, it comes out at about that sort of size, about 20 to 30 nanometers as well. So there seems to be some kind of very efficient system here, which we don't know how it's working. And if I was giving this talk in 3 months' time, I'd be able to tell you about the wonderful time I'd had at Diamond in November, where we're going to use the NanoProbe hard X-ray microscope, which has about a 25 nanometer resolution. We can scan across the cells. We can not only count the number of ions in here, so we've got a good estimate of the intracellular pool concentration of ions. We will also be able to do X-ray spectroscopy on the nanocrystals and tell you what they're bound to as well. And 25 nanometers is about the resolution of a, so you can actually see the periplasm is about 30 nanometers across. So we should be able to see whether things are in the periplasm or inside or outside the cell. So the population is the next level that we looked at. And this work by my colleagues Dov and John came out a few years back, where they looked at the zinc system as well. And previously, bacteria had been modeled like this, with a sort of efflux and an influx and an efflux out. And if you measured the balance between the two, that told you allegedly what was going on. When you started to actually put in all of those components of the system, and also started to knock out some of these, so it affected the behavior and the rates of these things, there were two things that turned up that had to be modeled. One, that you had to have a decent internal zinc reservoir. So there are always zinc ions in there, and these won't be free, right? They'll be bound to something. So they might be in metalloproteins or in chaperones or some kind of storage mechanism. The other, and that was the first, and that's intriguing for the copper-silver system, because with copper, of course, you have to have homeostasis because you have copper ions, whereas silver is lethal, so you just have to get it out as much as possible. So whatever silver is binding to within this pool of stuff, it's going to be binding. That might give you clues to its bactericidal effects. Because if you read in the literature, it says, well, it affects the DNA, it affects the membranes. Yes, it does. But it also sits where copper normally sits, right? Like a plug, like we saw in SIL-F, where it just sits in the binding site and doesn't do anything because the energy levels are all wrong for electron flow. And so you can take, standard sort of mathematical model here, and doing this, they were able to show that you could start to model this. But there's one piece of perturbing information, was that system only worked if there was heterogeneity in the expression of these things. So some of these things would have a lot of these zinc things already expressed in the system, irrespective of whether there was a zinc challenge, and some would have none. And this is a strategy called bet hedging. So the system essentially is hedging its bets against an unknown future, where some of the population have overexpressed, proteins on the outside, and therefore able to cope with the challenge, and some do not. And there's obviously a big heterogeneity, and we want to know is zinc, where it is, and we can do sort of some cell sorting. And the crucial thing about the take-home message is this: because there is a difference here in this portion of the curve, that shows that actually these things are heterogeneous in their expression of these across the population. So you've got heterogeneity at the molecular level, you've got heterogeneity at the cellular level, And we see heterogeneity now, which we can model at this sort of level as well. And these sort of bet hedging mechanisms basically protect bacteria against, as I say, against an unknown future. They don't know what challenges they're going to get in the wild, but all you need is one bacteria to survive, because these things under the right conditions will double every 20 minutes. So you can throw away 10 to the 1 in you can only have 1 in 10 to the 9 survive, but eventually it will outcompete everything else, which is why you get resistance with antibiotics, because you've killed everything else, so only the resistance survives, even though initially they may only be there in very small amounts. And the final level we want to look at, really, and this is the bit that we've only really started to touch, is the spatial level. So if you've got heterogeneity at the at the population level, then obviously, how does it affect this thing when it is in a biofilm? When you are when the three-dimensional arrangement of these things is important, because a lot of the sort of microbiology that I do is done in a flask in the lab, and that's nice warm environments. 37 degrees, lots of nutrients. Well shaken. E. coli will double every 20 minutes. That's fine. Out in the wild, it's going to be stuck to something. And so the 3D arrangement of how these things are important, and looking at systems like Pseudomonas, you've got a core of, should we say, less hardy cells and more resistant cells on the outside. And these things, certainly to antibiotics, can be 1,000 times more resistant than the cell. And again, we've not really looked at the metals, but we know they're there. And we know that the metals are out there in the environment. So very quickly to active matter. 3 things I think should notice. That firstly, the bacteria are obviously active matter. These are systems well away from equilibrium, because as any first year biochemist will tell you, if your bacteria reaches equilibrium, it is dead. So how we take the sort of results and things from that sensibilities from that sort of out of equilibrium systems, I think is still very much an open question as well. We need to, if you'll consider the insides of these things and what's going on, if you want to describe things at the top level as well. And, does the bacteria of choice cope with heterogeneity gives a clue to how these active systems adapt, because adaptation, in biology, that's evolution, that's what's going on. But how do you put that sort of description together? So those are my thoughts, really sort of throwing that out on that system. So why is these sort of system important? Because, looking at sort of these sort of esoteric sort of ion binding systems is all very nice, and you can do some nice biophysics, and I've had a lot of fun doing this project, but actually there's a really, really important reason for this, and which of course is we want to look at this from a perspective of antimicrobial resistance. And the problem of AMR is big and very real. And so we are looking for new antibiotics, and we're looking for new antimicrobials. And certainly silver seemed like a very, very good idea, because, as I say, it kills bacteria. You used to be able to get it impregnated in deodorants and socks and all sorts of things. And so there's lots of silver now actually in the environment as well. And that sounds great, because then you can just kill bacteria, and you don't need to use antibiotics, and you can use those in the clinic. There is a problem, and that is that silver resistance and copper resistance usually are on mobile genetic elements, plasmids, which can be passed around bacteria. They aren't the only things on those plasmids. There's also typically a whole host of antibiotic resistance as well. So you select for silver, you select for the whole pack of cards. And so we started to look in the environment in the clinic about this. So we went up to the QMC Path Lab, Queen's Medical Centre in Nottingham, and asked them for their most drug-resistant, antibiotic-resistant bacteria they fished, E. coli, they fished out of patients, right? And we got about 8 of these, and we went and sequenced the plasmids, and they are all metal resistant. Even those things that we're pretty sure they've never seen silver or copper in their life as well. So where are they coming from? Well, my colleague John Hobman is a real microbiologist, as in he goes down the slurry pit on the university farm with a bucket and comes back with stuff to measure. And he couldn't work out why he was getting lots and lots of copper-resistant bacteria in this slurry pit. Okay, antibiotic bacteria, it's in the environment. We don't put it into cows anymore. And then he had a look at the colour of the foot bath the cows were going through. It was blue. It's copper sulfate. And it also contains zinc sulfate. Where did they put the used foot bath? They put it in the slurry pit. When you've incubated everything there for 40 days, because we think that's where the multi-drug resistant bacteria die off the most, where do you put the slurry? You put it on the fields. What do you grow on fields? Food. What do you eat? Do with food? You eat it. Okay, hopefully you've cleaned it. Right, so it's already out in the environment. We can see it in the clinic as well. And this, again, is a problem of disciplines in the fact that for years you had food microbiologists who worked on cheese and keeping pathogens out, medical microbiologists who worked on basically what came out of a patient and stopping them from dying from it, environmental microbiologists. And the trouble is, bacteria aren't any respecters of discipline. They will quite happily go through all of these things. So a lot of this has been what they call one microbiology, is trying to draw together many of these lines here as well. And as we start to, and then as we've started to look at this, then we have to see that over this vast number of sort of results potentially on E. coli as well, that you've got these levels of description. And when you look at something allegedly so simple like iron binding, it seems to be operating heterogeneously over levels of description as well. So the way sort of put together, and I've been really quite interested in a lot of the machine learning talks today. I mean, you think you're working on polymers, but I'm going to see some quite a large amount of overlap with some of the stuff that we want to do. It's how we actually now start to describe this and put it together. So we are starting to flay a little in the dark about our descriptions here. But I think we've now starting to this is now starting to coalesce into something which we can do. And I think we can now start to model those things across the level, because at the moment, the grant applications, the modeling bits goes, oh, and we're just going to model it across the levels. And for some reason, referees are quite resistant to recommending funding on that little amount of information. Yeah. So rounding up, on a wider level, there is E. coli is a good system to look at because we understand it, that you can make mutations. You can do things in the lab, within about a week, you can, mutate the hell out of it, knock things out. It's great. It grows overnight. However, it's only one of a set of nasties out there. Many of these here, some of these are gram-positive, some of these are gram-negative. And the World Health Authority and the like are very worried about these organisms. So what we can actually do on E. coli, we're hoping we can actually transfer across to these other things as well. And with that, I'm at just coming up. Yes, 5 minutes before the end. I'll leave it there and I'll take your questions. Thank you. Yeah.
I think talk about how still poison. I mean, it's still poisoned by outspeaking other good iron. Does that mean the whole system still depends on concentration of other iron? If you give something silver and lots of good iron, then silver's less toxic than if you have a deficit of the.
It might just be sitting there like a plug. Years ago, somebody sold an iron channel, but from the nerves, but with plutonium. And you can see why plutonium affects your nerves, because it just sits there like a plug on top. So it could be doing that. The other thing is silver ions like to react with anything. One of the big problems we've had doing the in vitro work was finding a biological buffer that didn't precipitate. Phosphates, chlorides, all these things form insoluble precipitates. So it just gets in there, and it'll bind to the phosphates on DNA. it'll bind to bits and pieces of cell membranes, and it'll just destroy its integrity. It's a reasonable amount you need, about 50 micromolar, and to get it in there. And the resistance mechanism pushes that up to about 600 micromolar. But yeah, it seems to be a combination of basically just planting itself there so nothing happens, and also at being quite reactive. Yeah.
Did you find it filled with less to us?
Yes, it is. Now that's
Why? That's weird. I mean, we've got DNA and
yeah, absolutely.
I
Yeah.
think.
Yeah, I mean that's one of the things that has perturbed us. I mean, as I say, you can drink silver colloid, and people did it as a prophylactic, especially during.
I would do that, but yes,
Hmm?
I'll pass on that, but yes.
Yeah, and as I say, beyond the fact that they went blue, it has no massive ill effects on them. So, one of the things we are looking quite actively now we have the techniques, is actually how silver works, because when you actually go through the literature on this, it's like, yeah, it kills, yeah, and we think it's the membranes, and we think it's the DNA, and of course that gets repeated and repeated and repeated, and nobody actually looks at the original thing because it was sometime in the 60s.
I mean, do we have silver pumps?
No, but you'll have copper
Okay,
homeostasis, and copper's in a small iron, quite important, your cytochromes. Yeah, so yeah, what it's doing, and yeah, I mean, you can get a condition called aguria, which is when you have huge amounts, and you won't die, but you'll be quite ill. Yeah.
Thank
Yeah.
Could I ask about the bet hedging
Yeah.
thing? So where does the heterogeneity come from? Is it just random expression levels, or is there a more controlled mechanism?
That's a good question. At the moment, we're thinking it's just sort of stochastic, that, some cells, on the whole, there is a certain amount of noise in these systems anyway, and some cells just express these things slightly more and some don't. It also gets down to the regulation, the control of the promoters as well. If the repressor protein does not bind particularly tightly, you're going to get some on-off and some expression as well. So that's a nice open question. Mm.